So we're going to move on to the next section, which is about biomarkers, which is relevant and thinking about how biomarkers might help us in these clinical trials to know whether drugs are available or not. So we move on to the next slide, biomarkers of FTD, and I'll hand over to Emily, Misha, and Etana, who are going to go through this section.
Okay, hi everyone, my name's Emily, and today myself, Misha and Etana are going to be talking to you about biomarkers in FTD. So firstly, what is a biomarker? So a biomarker is a measurable characteristic that helps us to understand what is happening in the body. So for example, blood pressure, but in the case of FTD, we'll be talking about imaging and fluid biomarkers. Next slide, please, John. So as I said, I'm Emily and I'm the imaging RA at UCL and I work in FTD. And today I'm going to talk to you about how we can look inside the brain to investigate FTD and how we can look pre-symptomatically, i.e. in the time period before symptoms begin, and then why this is useful. Next slide, please, John. Oh, okay. So what is an MRI? I'm sure some of you will have had an MRI before and will know what it is, but MRI stands for magnetic resonance imaging. And in simple terms, it's a procedure using magnetic fields to get a signal from inside the head that allows us to have black and white pictures of your brain. Next slide, please, John. So how can we use this to help us understand FTD? Well, FTD causes loss of brain tissue and neurons in a process called atrophy. And we know from research and by looking at patients in a clinical setting that individuals with FTD tend to have specific patterns of brain atrophy, with the main areas affected by volume loss being in the front and the sides of the brain, known as frontal and temporal lobes. However, we know that FTD is a complicated condition and can have lots of different symptoms associated with it, with different parts of the brain controlling different actions and processes. And therefore, the reason why we see different symptoms is because different brain areas can be affected. For example, if language areas of the brain are affected, we then see language symptoms. We can categorize into different diagnoses of FTD, such as language variants, including semantic variant PPA, and non-fluent variant PPA, and behavioural variants such as BVFTD. Here you can see a patient with more behavioural changes compared to a patient with language symptoms, highlighting the pronounced areas of atrophy as seen from above. You can see that in behavioural variants, atrophy tends to be more localised to the front of the brain, whilst language can be typically localised to one side of the front of the brain. This can be helpful in research as it helps us to group patients together by their diagnosis. You can either look at the images such as these for qualitative research, or you can use measures to quantify the volume differences. Next slide, please, John. For example, we can extract volume data by using specific imaging packages to give us information about different brain structures, like the ones shown here, allowing us to compare volume loss across diagnoses and patient groups. As you can see from this image, distinct brain structures can be seen by different colours, and we can group these in different ways to quantify different brain sections. We can now even divide brain structures into even smaller sections, looking at specific regions of brain anatomy and relating them to the progression of the condition. An example of the subsegmentation is shown on the right, in the right image, which is of a thalamus. Next slide, please, John. We can also image the white matter connections between brain regions and help us understand how regions communicate. We can do this through diffusion tensor imaging and tractography, through specific MRI sequences and imaging software. Next slide, please, John. Similarly, we can use techniques to also look at the differences by genetic mutations, with progranulin mutations tending to show a pattern of asymptomatic atrophy, C9orf72 mutations showing generalized atrophy across the brain, and MAPT mutations typically showing atrophy in the temporal lobes, so this would be at the side of your head by your ears. Similarly, differences in pronounced white matter structural decline can be observed between the three genetic mutation groups, with MAPT carriers showing poorer white matter connectivity in tracts connecting the frontal and temporal lobes than progranulin, and with progranulin mutation carriers showing reduced white matter connectivity in central white matter tracts in the MAPT carriers. Different mutations appear to have different typical symptoms, and this is because the condition is affecting different brain areas. So, for example, MAPT carriers tend to show problems in emotion regulation, and with the temporal lobe being a key area in emotion regulation, we often observe atrophy in this region. Next slide, please, John. It is also helpful to study MRIs of individuals with genetic mutations because it allows us to compare brain volume by groups of people at different stages of the condition. We can detect changes before the onset of symptoms using accurate measures that can quantify where and when changes in the brain occur. Therefore, we can have accurate measures way before the onset of symptoms, and this can help in the design of future clinical trials. We can also quantify the changes that occur in the brain over time, whereby we follow individuals over multiple visits and track how much tissue is lost and measure the rate of brain deterioration and disease progression. I'm now going to hand you over to Misha, who's going to speak a bit more about PET imaging.
Hi there, thanks Emily. So I'm going to talk about a different type of imaging biomarker in FTD. So the participant's experience for PET imaging is largely similar to an MRI scan in that you enter a tunnel-like scanner and pictures of your brain are taken. However, there are a few key differences, some being in the scanner itself. It's a different type of scanner, it's much wider and it makes much less noise than an MRI scanner. But a key difference is that just before the PET scan, somebody will inject the participant with something called a radio tracer. So PET imaging stands for positron emission tomography, and this means it counts the signal emitted by the radio tracer to form images instead of about brain structure, about brain function. So on the next slide, we see what an MRI scan looks like from Emily. It's a black and white image that tells us a lot about the structure of the brain itself. But if we see the next image, this is a PET scan, and on top of that MRI template, so looking at the structure of the brain, we've added some information about the function of the brain. And the function of the brain is informed to us by a protein of interest. So on the side of the image, we always have a color bar, which represents the intensity of the signal that we're seeing from the protein of interest in the brain, which is collected by the scanner. If we go to the next slide, we can see what these images inform us of. So we have different areas of activity in the brain of the protein of interest, depending on where the radio tracer or the injection is emitting the most signal from in a PET scan. We have areas of high activity, areas of medium activity, and areas of low activity. And this might correspond to areas of lots of protein, areas of little protein, or areas of lots of function of a particular protein versus little function. On the next slide, we'll have a look at some of the proteins of interest that we look at using PET imaging. So there are multiple things in the brain that we think are involved in neurodegenerative diseases like frontotemporal dementia. And 3 of these include protein accumulation, inflammation, and connectivity. If we think about protein accumulation, most neurodegenerative diseases, so diseases like dementias, are caused by the gathering of faulty proteins in the brain. And in frontotemporal dementia, we know that these are the tau protein and TDP43 in most cases. And we can use PET imaging to inform us about protein accumulation and whether there is a particular gathering of tau in different areas related to the disease process. So, for example, with PET imaging, we do see increased amounts of tau in the brain in areas related to the disease process in FTD. We can also look at inflammation in FTD. If we think about inflammation in response to injury in the body, where we have an injury in most areas of the body, for example, a paper cut, inflammatory cells will respond to the site of injury and try and mend the site of injury. And the same might be happening in dementia. So we look at inflammation in the brain to see whether it corresponds to areas of the disease. And indeed, when we look with PET imaging, we see increased inflammatory signal in the brain, suggesting either that it's part of the disease or that it's trying to respond to the disease process itself. And either of these things might be helpful going forward in identifying treatments. And the third thing that we can look at in frontotemporal dementia is connectivity. So when we lose brain cells in frontotemporal dementia and other dementias, we lose synapses. Synapses are the connections between brain cells, and they're super important for the communication between brain cells and activity. And what we suspect in frontotemporal dementia is that connectivity, or the number of synapses in the brain, is reduced. And this is indeed what we see with PET imaging. So we see a reduction of synapses in areas related to the disease process. And all of these things might be really helpful in informing us of what therapeutic targets we need to go forward and identify. So these are important in helping us to stratify participants, and that is to understand what type of disease they have during life, as the PET imaging is the only way to look inside the brain during life. But they also might help us to identify treatments that we could use going forward to treat frontotemporal dementia. And I'm now going to hand over to Aitana, who's going to talk about fluid biomarkers in frontotemporal dementia.
Hello, good morning. I hope you can hear me properly. And sorry, because I was not able to connect my camera on. So I'm Aitana, and I will talk you a little bit about the fluid biomarkers and FTD. So if you go to the next slide, please, John. So what is a fluid biomarker? My colleague Emily said before the definition of a biomarker, but a fluid biomarker is indeed something, in our case, is a protein, a lipid, or genetic material that we can find in a body fluid, and we can measure to know what is happening in the brain and what is happening in our body. We can use the fluid biomarkers to help the diagnosis, but also to help monitoring a therapy. So please, can you go to previous slide? Sorry. Where do we measure these fluid biomarkers? We can measure them in spinal fluid. This is the most used fluid mentioned because the spinal fluid is the window to the brain. So the proteins or the molecules that you would find there can tell us what is happening in our brain. However, the method to extract the spinal fluid is a little bit invasive. So now the research is moving on to find fluid biomarkers in other fluids like blood and urine. The good thing with blood, it has been shown that we can find brain molecules or brain markers in it, and the method of extraction is quite easy. If you go to the next slide, please. So what can we measure with the fluid biomarker, and what can this tell us about FTD? So we can know about the brain pathology, we can know about the neurodegeneration, and we can know about the cellular dysfunction. So regarding the brain pathology, if you go to the next slide, and it has been mentioned before, FTD is a very heterogeneous condition, and a part of the cases has a genetic cause, and we have three main genes that can cause FTD. And in the case of the biomarkers, unfortunately, we don't have a biomarker that can tell about general FTD, but fortunately we have biomarkers that can tell us about its different mutation. So in the case of the C9 operinephrine 72 mutation carriers, we have the dipeptide repeats. In the case of granulin, we have a molecule that is called progranulin. And in the case of the MAPT, we have tau. So this protein has been studied in fluid biomarkers. However, any of them is still validated to use as a diagnosis. If you go to the next slide. In the case of the dipeptide repeats, it has been shown that they are increased in CSF of both presymptomatic and symptomatic mutation carriers, and it can tell us the difference between the presymptomatic and the symptomatic ones as well, and is not present in the non-mutation carriers. If you go to the next slide, please. In the case of progranulin, this molecule is decreased in the granulin mutation carriers. So what else can a fluid biomarker tell us about FTD? It can tell us about the neurodegeneration. In this case, we have neurofilament light, the general marker of neurodegeneration, and unfortunately it's not specific of FTD, but this is a good marker that can tell us when our neurons are dying and is increased before the symptom onset. So why they use it in other dementias as well as in FTD. If you go to the next slide. And finally, a fluid biomarker can tell us about the cellular dysfunction. And here we have different candidates. In the case of neuroinflammation, we have the DFAB, or in the case of lysosomal dysfunction, we have lysosomal markers, and for synaptic dysfunction, we have different synaptic proteins that can tell us about that. All of these markers are now being studied in the research, and hopefully we will find one of them that can tell us more about it. So if you go to the next slide, in the case of DFAB, studies have demonstrated that is increased in the granulin mutation carriers. And I think that's all for me. Thank you very much for listening.